Hi, good morning, everybody. So this is a little bit of a new research topic for us. So endosymbionts seem to be very widespread and are thought to be carried by nearly half of all arthropod species. And they can be important for their biological functions and their life cycles. But these interactions between these heritable bacteria can either be positive, can have no effect on the host, or they may have a negative effect on the host. So some of the effects that we know that they may have on their host are such as cytoplasmic incompatibility, whereby an infected male needs to mate with an infected female in order to produce fertilized eggs. So if an infected male mates with an uninfected female, no offspring will be produced. They may undergo feminization, whereby females may be produced, all females will be produced, even though some of them may be chromosomally male. Male killing may happen, whereby the total number of offspring is reduced, and then those that are produced are mainly female. Parthenogenesis, whereby females may produce only female offspring without the fertilization of eggs. Parasitoid defense, whereby the arthropod gains some defense against things like parasitoid wasps, infecting them. And pathogen blocking, whereby the hosts may be resistant to certain RNA viruses. So as I mentioned, these endosymbionts may appear to also have no effect on the host, as is the case with this leafhopper, whereas we can't see any effect in the actual host itself, but once it sucks sap from a plant, the endosymbiont is transferred and is a plant pathogen. The relationships between the host and the endosymbiont may also be situation dependent. So in this case, the aphid has a clear advantage when it has the endosymbiont because it is resistant to these parasitoid wasps parasitizing them. But in the absence of these wasps, having the endosymbiont can be quite costly for their reproduction and their longevity. So in this case, it's not an advantage if the wasp is absent. There's also a number of mechanisms for transferring the bacteria between species or individuals, and I'll just mention a few of them. So this is an example of horizontal transfer, whereby this whitefly may suck sap from the leaf, and if it is infected, transfer that endosymbiont to the leaf. And then another species of whitefly, for example, may suck sap from that same leaf, and it may then pick up that endosymbiont. And this is an example where we see often unrelated host species having the same species of endosymbiont. Another example is if the host has an endosymbiont. So this phylogeny is showing the relationships between the endosymbionts. And I've marked the host, which in this case is a caliphorid, and the species above it are all parasitoid wasps, which use that caliphorid as its host. So in this case, the endosymbiont has been transferred horizontally between those species via the host. And then, of course, we all know that it can be passed down through generations if they're heritable bacteria. So it's passed down maternally from generation to generation. So this is just a cladogram of some of the most common endosymbionts. So we can see they infect a number of arthropods, and they may have a number of manipulations or effects on the phenotype. And this research is continually showing us new effects it may have, as is the case in 2024. There was cytoplasmic incompatibility was a new record for Rickettsia of its effect on phenotype. So now the interesting part, what this does in Diptera. So it does seem to be quite widespread in Diptera, but what we know is mostly from the blood feeding groups or those of economic importance. And of course, this makes sense and is of great interest to us. Don't worry about all the detail in the table. But what I do want to highlight is that in mosquitoes, it's been shown that the presence of endosymbionts can, for example, is linked with lower infections of dengue in the mosquito itself, so that results in then lower transmission to humans. Or it can increase or decrease the susceptibility to insecticides, which is of course important if they're a pest species. But what we don't know is the presence or frequency of these things in lesser known groups, such as the spider flies. So spider flies are really small, or relatively small family, and is characterized by having this small head and usually quite an arched thorax. And this earns them the common name small-headed flies. The other common name is spider flies. So for part of its life cycle, they may use a spider as their host. And you can see there's a very unfortunate salticid spider there, which they then use as a food source during their larval phase and then emerge from that spider to become adults. And as adults, they may be important pollinators. And in South Africa, there's a number of species of flowers which may rely on these flies for their pollination. But other genera in the Acroceridae, we think, may not feed at all as adults. So how did we come about this? What we were actually doing is a taxonomic revision. So we weren't looking for endosymbionts at all. What we were trying to do is revise the genus Silodera, which are these cute little pollinators. There's 9 species in the genus. And as many of us do, we were trying to do morphology and add some barcodes to that to link males and females and so on. But what we found when we generated our first barcodes were, okay, we found Silodera barcodes as we expected, and then we found Wallenbachia. So we didn't get fly barcodes where we expected to find Silodera hesse, we found Wallenbachia, and then for 2 more species we found Rickettsia. So Wallenbachia has been reported in a single Onchodes species before, but Rickettsia, this is the first report for the family anywhere in the world. Our sample size at the moment is quite low, so we only did a few individuals because this was meant to be a revision originally. But we did find these endosymbionts in the species where we found them. They were in every single individual we sequenced. And this includes males and females. So we also screened a number of other Acroceridae genera for which we had representatives. And the only other genus that we could find an endosymbiont in was Thyllis. And then I just want to point out a few things. So it seems like, so the yellow, if you remember, is Wallenbachia, and this was Silodera hesse that we barcoded. And those 2 individuals underlined came from the same sampling event, but it does appear that there's at least 2 species of Wallenbachia or very closely related strains. In Rickettsia, there's very obviously 2 species here. So the one was is exclusively found in Silodera confusa so far, and the other species of Rickettsia seems to be more widespread across different species of Acrocerids. So then the last thing I want to mention is that for this revision, we looked at many specimens, and they're overwhelmingly male. So for Silodera confusa, I looked at 107 specimens, and 90% of those were male. And for Silodera hesse, I looked at over 80 specimens, and 80% of those are male. So if we think about what these endosymbionts typically do, not in all cases, but they do tend to favor females. So what's happening here, we have no idea at this stage. I cannot rule out the possibility that it's just a sampling artifact, but it does seem quite extreme, and it's across collections, across decades, so who knows what's happening there. And then our next steps are to screen more species, genera of Acroceridae, and then for species which we have a number of individuals, we'll try and see if we can determine frequencies of these symbionts within the groups. Thank you.
Okay, are there any questions for Genevieve? Okay, this one. That's it. Someone in back as well.
Hi, thank you for your talk. So what kind of sequencing did you do? Do you have access to these genomes so that maybe you can actually look for the genomic composition and possibly infer whether they could be doing CI or male killing or even be provisioning any kind of nutrients?
So at the moment, well, the data that I presented is just barcodes. So it's just CO1, the standard barcoding region. But in the future, we hope to do some more next-gen sequencing. So we'll be able to determine more once those come back.
Somebody in the back?
Thank you for your presentation. You have a slide whereby you are saying the rickettsia is horizontally transmitted. Is it only horizontally or it can be vertically?
Rickettsia?
Yeah.
What are you asking? Is it only?
Is it only horizontally transmitted to the insect?
No. So they can definitely be they're heritable, so they can be transmitted between generations, from mother to offspring, and horizontally between, I guess horizontally, if there's a host association or something.
Okay. Thank you.
The other question there in the back? Last.
Genevieve, obviously you aren't familiar with spider taxonomy, but it would be interesting to look at what the host range of Acroceridae is on a broad scale globally, and actually to plot that on the phylogeny of spiders too, to actually show whether they are focusing on more advanced families phylogenetically in terms of spiders or the primitive families. Most of the records I know of are relatively advanced spiders, so jumping spiders, clabionads, and so on. So it would be interesting to plot that as well.
Yeah, all those things are very interesting, but unfortunately, Acroceridae are not very regularly collected. They're quite scarce in collections, especially modern ones, so that we can have molecular data. So, and what we know even less about is their hosts. So I think that information is quite scarce, especially for South African taxa, but it would be a very interesting exercise if we could do it.
Okay, Ben, last question.
Thank you, Jen. I'm just a bit confused about how you managed to get bacterial barcodes out of insects. How did you manage to, am I just confused, how did you manage to separate the two?
We didn't. We tried to barcode flies, and what came back was bacterial DNA.
Even though
it was absolutely not intentional.
Yes.
It's just a fantastic result by accident.
Do you know how that might have worked? Like, is there some kind of similarity in that particular section of mitochondrial genome that just happens to overlap with these kinds of bacteria?
No, I'm not sure. Sorry.
Okay.
Good. Thank you. Well, basically, they have the same protein genes, and the primers, they do adhere to that one of the bacteria as well. So it's just a matter of chance with Sanger sequencing. Where it sticks first, it will get amplified. And in some cases, it's the bacterium. In other cases, it's the fly. So if you really want to separate both, you need to have some next generation sequencing technique, and then you will get both. You will get both the host and the bacterium.